Hello, my name is Sydney Horniker, and I did my final presentation on the behavioral genetics of the honeybee. So just as an introduction, honeybees are the model organisms for behavioral genetics studies. They're extremely social organisms, so it allows them to form this model behavior. Their genetics and the evolution of their genes has aided in the expression of their social traits. So over time, they become much more social and they interact with each other much more than they had. Um, the honeybee genetic sequence has fully been discovered, allowing the connection between genes and social traits. So now it is possible to dissect those genes and determine what social traits correlate directly to them. This allows genes to be able to be traced back to worker bees' specializations and why they have these specializations. In this image, I am showing the worker bees and what their typical lifespan is and what they're doing. Just as an outline for what I will be talking about, I will be discussing the methods and processes of tracing a honeybee's genes to their specific jobs, the problems that may occur with the methods that are commonly used, and the genetic architecture and task specialization of honeybees. And this image is just a breakdown of, where they get their color, and basically that's all. So the methods and processes that are used commonly is reverse genetics, forward genetics, and linkage mapping. This is a super complex version of a linkage map. So reverse genetics is basically just a connection between the qualitative and the quantitative genetics. This is done through microarrays and needing a complete genetic sequence. So without the complete genetic sequence, this is impossible. So they first start with a list of genes where they already know or it is presumed what the function is in a different organism. So it is not in the organism that you're looking at. However, it is a different one that would closely relate. Then those genes are tested for the involvement in the suspected behavior within the organism that you're looking at. So in this case, you would test the involvement of the suspected behavior in the honeybee. This allows conclusions to be made across taxa. So, you're able to take this organism, relate it with this one, and decide both of them have the same genetic sequence that causes this suspected behavior. Therefore, they are similar. It should be across all taxa. In these images, I have a little identifying the honeybee. This is what a honeybee looks like. There's a bumblebee, a hornet or a wasp, and a hoverfly. And this is the taxa of the honeybee. Moving on, forward genetics is basically creating a hypothesis with or about the activities that correlate directly with specific genes. But by testing this, you can determine the validity within your hypothesis. The forward genetics maximizes the chance of detecting differences in gene expression genotypes associated with differences in behavior. In this image, you are going to start with a phenotype and then you're going to screen a collection of mutants. So you would screen your organism and therefore you'd be able to identify the gene or the genes causing that specific phenotype. So it's basically working from what you already know to what you don't know and what you want to figure out, just as a normal science experiment. The final option listed in this paper was linkage mapping. And this is just a quantitative analysis of the genetic effects on behavioral traits. It helps reveal the presence and the location of those genes through these maps here. Quantitative trait loci, or QTLs, contain more than one gene. Sometimes they only contain one gene, and it leads back to one specific phenotype variation. QTLs are used to cross-link behavioral traits with presumed genes. So that is shown here and here. Issues and problems with the aforementioned testings. In reverse genetics, your researchers are really restrained to only the genes that have been conserved from other model taxa. And given that honeybees are extreme model taxa, or they're an extreme organism, they're an extreme model organism, I should say, it makes it really hard to compare it with other taxa. Honeybees are extremely social. I would go as far to say as the most social organism. So their gene and the gene expression that they have is very different as compared to another organism. So reverse genetics doesn't work very well with honeybees. Forward genetics, by hypothesizing in advance, you're creating a large amount of tests that need to be performed, which often leads to false discoveries. There's a lot going on with the making a hypothesis and determining what genes go with what phenotype. Therefore, there's a lot of backtracking and miscommunication that can occur there, meaning that it's not a very reliable testing method. The final option, linkage mapping, as you've seen in the first two, or the two images of the linkage maps, they are clustered and they are confusing, and there's a lot going on there, so it makes the data extremely hard to interpret. So it's just not commonly used at all because there is so much to interpret. Next, I'll be talking about the genetic architecture and task specialization. These next bullet points are the subcategories of the genetic architecture and task specialization. So I'll be talking about task stimulus and task threshold, behavioral overdominance, the physiological and genetic basis of the task thresholds, hygienic behavior, and dance communication. So in task stimulus and task threshold, queens typically mate with 10 to 20 haploid males, these males producing their sperm clonally. This allows for about 3 times more genetic difference between these subfamilies or the patriline's. So in these different subfamilies, the workers tend to behave differently based on their task stimulus. And the level of the task stimulus experienced by an individual worker is influenced by the activities of other workers and by unequal numerical strengths of patrilines. So this being said, subfamily 1 may perform one task, and there may not be a lot going on in that task, so they perform that task and they stick to that. Whereas subfamily 2 may perform a different task. There's not a whole lot of cross working due to it working for them. Yeah. And when a numerically large subfamily starts to engage in a task, it will reduce the level of stimulus within the colony. So basically what I just said, if this subfamily is working on this task, let's say subfamily 1 is working on a specific task, subfamily 2, 3, and 4 basically don't engage in it at all because it's already being taken care of. So that reduces the level of stimulus for the colony for that specific task. All workers typically have a diverse behavioral set of skills where each has its own threshold. Workers may abandon one task should their threshold for another be exceeded. So if they are performing one task and another is better fitted for them, they will leave their current task to move on to their other. Next, I will talk about behavioral overdominance. This one is a colony level phenotype. It's strongly influenced by a relatively small proportion of workers. There's 2 forms of it. The first one is if a colony's need for a task is taken care of by a small amount of workers who have a low threshold for said task, the colony reflects the phenotype of the minority low threshold workers. The second one is if the activities of a small amount of workers lowers the task threshold of other workers or may increase the task stimulus that the nest mates perceive. And then here is just a little diagram showing that if this, let's say genotype A or genotype 1 increases, the other two are then going to decrease in response. So physiological and genetic basis of task thresholds. The tasks and duties are entirely based on the age. So whenever honeybees are young, they typically stay within the nest. They guard the nest. They don't perform tasks outside of the nest. But whenever they reach about 3 weeks old, that's when they start their foraging task, and they'll basically be doing foraging tasks for the rest of their lives. This is entirely based on their exocrine and their endocrine glands. This is making sure that they have the appropriate glandular secretions to do their tasks at their respective ages. So once they have matured enough through their exocrine and their endocrine glands, they're able to then go on and perform other tasks. As they get older, their wax glands begin to learn the scents of flowers and the locations of those flowers after they begin their foraging process. So they're already in their foraging process, and the wax glands are then learning these flower scents and the location of those said flowers. The rate at which bees progress from hives to foragers is caused by social interactions with other workers. These social interactions with other workers typically accelerate the production of the juvenile hormone, and producing more juvenile hormone accelerates the bee's likelihood of being a forager. So it just makes them go to become a forager much faster. Nurse workers tend to have lower levels of the juvenile hormone because they don't often leave the nest. A continuation of that, the juvenile hormone is an important factor in regulating the behavioral development of workers. So just like I just said, higher levels of that juvenile hormone will increase their likelihood of becoming a forager. It'll make them become a forager faster. And the juvenile hormone is shown here. Um, the age at your first foraging is genetic. Okay, ignore that. Patrolines with slow development of the genetic hormone are likely to continue to stay in the task or in the nest and perform those nest-based tasks, such as guarding the nest. And the subfamilies of patrolines with rapid development of juvenile hormone, are going to begin those foraging tasks and working outside of the nest. It is suspected that many instances of task specialization are emergent properties of genetically based variance and rates of behavioral ontology. So moving on to hygienic behavior. Hygienic behavior is determined as a colony level phenotype, and this is based on how fast the colony cleans out the brood cells, which is dead larvae and dead pupae. On an experiment basis, 100 brood cells would be provided to a colony. If the colony removes all of these brood cells in 48 hours or less, it is safe to assume that their colony is hygienic. However, if it takes them more than 48 hours, they're considered non-hygienic. By finding the genetic markers to identify hygienic colonies, it would be extremely useful for selecting colonies for further breeding. Um, a, then moving on, a portion of the colony must be middle-aged in order to express this hygienic phenotype. A middle-aged honeybee would be around 2 to 3 weeks. Middle-aged workers are the only ones that are able to uncap the brood cell and remove the infected pupae for disposal outside of the nest. So they would take that outside of the nest, get rid of it, and then as that process continued, they would all be removed, considering the nest hygienic. It is suggested, it is not proven, that the uncapping and the removal of these pupae are under separate genetic control. So the uncapping is of one and the removal is of another. And they're each controlled by a single unlinked Mendelian locus. Hygienic behavior is also based on the stimulus threshold of reactions to the odors of disease-killed brood cells. So a colony that has an increased hygienic behavior can determine the difference between a healthy and a disease-killed brood at a much lower concentration, a much lower concentration of scent than that of a line bred with poor hygienic behavior. And then when exposed to equal levels of odor stimulus, both a non-hygienic colony and a hygienic colony, the hygienic colonies experience a much stronger electrical signal in the antenna lobe, in the antennal lobes of their brains. This creates high levels of the neuromodulator octopamine, which is shown here. High levels of octopamine are extremely crucial in the behavior of hygienic behavior in honeybees. And this is possibly due to its necessity of information of the olfactory memory in honeybees and organisms alike. Finally, I'm going to talk about dance communication. When returning from a foraging mission, a foraging quest, I guess, a successful forager will perform a dance, and it allows the nestmates to locate where this profitable patch of pollen is. And basically, all they're doing is a dance, and this dance reenacts what the trip should look like and how to get there. So basically, she will perform what is considered a waggle phase, and she will stride forward as the other honeybees follow her while vibrating her abdomen. The alignment of her body indicates the direction of the patch. So if her alignment is straight up the comb, the patch is directly in the direction of the sun's current azimuth. If the alignment is straight down the comb, the patch is opposite of the sun's azimuth. And if the alignment is at 3 o'clock, the patch is at 90 degrees of the sun's current azimuth. Here it is shown what the waggle run looks like. It's a little back and forward motion with their abdomen. Continuing on the dance communication, the duration of this dance indicates the distance to the patch. So if the waggle dance is very short, it means that the patch is extremely close to the hive. However, if it is a longer waggle, then it shows that it's going to be a long distance from the patch. The bees that are following the dancer bee, for lack of better terms, can essentially estimate the distance to the patch by averaging out the duration of the waggle. So if it's a really long waggle and then another really long one and another really long one, they're just assuming that they're on for a long journey. But as the waggle gets closer, or as you get closer to the patch, the waggle becomes extremely hurried. So it's going to be a lot faster than it was initially. So whenever the dancer can't waggle fast anymore, it's getting too close, so it's physically impossible for the dancer to continue to waggle. It'll begin to do what's known as a round dance, and that is shown in this image, and it's basically just flying in a bunch of circles, and this just shows that they are extremely close, they're right on top of it. Here we are, here's my patch. The relative importance of genetics and the environment in shaping the dance temperament is unresolved. It has not been discovered yet. We're not that far. And as a conclusion, wrapping it all up, characterizing the genes related to social behaviors in honeybees will provide common ground for both functional and mechanical biologists. A very specific molecular focus, I should add, will help deduct the unknown pathways that link variation in nucleotide sequence to variation in high order phenotypes that characterize social breeding systems. In the future, they're hoping that the genes that control the major switches in caste determination and worker sterility will be determined. And here is my works cited, and that's all.